Okay, so what are we going to talk about today? Today we're going to focus on kind of the smaller part of the nervous system, if you will, right? So we're going to talk about what is the structure of a neuron and how does this relate to its function. Again, relating structure and function. How do we get a resting potential in a neuron, and how do ion pumps and channels relate to that? How is an action potential generated and conducted down the axon? Right? And how do synapses allow for communication between neurons? And there are some synapses that are electrical and some that are chemical. And we'll look primarily at chemical synapses. So starting at the very beginning, if we're looking just at one thing kind of in particular, we have kind of what's called the central nervous system and the peripheral nervous system, right? So when we're thinking about the peripheral nervous system, right, and the central nervous system, we're really talking about kind of like the brain, the spinal cord in humans. And this is really involved in the integration of sensory input, right? So you have the sensory input and then you have the motor output, and both of these are really happening through the peripheral nervous system. And we'll also look at some things probably later down the line where, because you're not always having to route your signal through the central nervous system. Okay, so we're going to take the sensory input, right? We're going to go ahead and integrate that in the central nervous system, and then we're going to go ahead and then output that in terms of some sort of movement or some sort of function, right? Using some sort of motor neurons at that point. So let's look at a neuron, right? So a neuron is made up of a couple of kind of like key things. So here are kind of two neurons. So one neuron on the left interacting with another neuron on the right. And so first, if you notice, right, you have this kind of cell body within which the nucleus and kind of all those organelles are contained. You have these dendrites, right? And so you can see they're quite small compared to that one very large axon. And these are going to go ahead and receive, right, the signals. Okay, so these are going to go ahead and receive those electrical impulses, right? In some cases, well, as we'll see with the synapse, right, they're also going to be involved kind of on the other end, right? So receiving these signals, and then we're seeing the axon, right? This is really going to go ahead and kind of conduct those signals that are input to the neuron, to the kind of the next neuron. And as you can see, we'll take a look at this in more detail, but the axons, they can tend to be very long, right? Where the dendrites tend to be kind of like more branched, right? And as you can see, there's kind of this single axon which is branching at the end. And as you also notice, right, you have this kind of myelin sheath that sometimes exists around axons. Oftentimes they're myelinated, and we'll kind of talk about that in more detail because that's going to go ahead and affect how fast an action potential can get sent down a neuron. So focusing on kind of dendrites are receiving, axons are conducting those signals down them, and then oftentimes axons can be very, very long, right? Sometimes, depending upon the size of the organism, up to a meter or more in length. Also, as we can see here too, right, as we're looking down at the synapse is where we're going to, well, you sometimes have electrical synapses, but we're going to focus primarily on chemical synapses. And at this point, you have these synaptic vesicles, right, which get released into the synaptic cleft, and then neurotransmitters are going to go ahead and bind, right, on these ligand-gated channels that exist on the postsynaptic cell. So that's something that we'll also go ahead and look at. So a lot of information here. So how do these neurons send information, right? So again, we're sending that information down the axon. So the information is going to travel down the axon to, right, so if this was the presynaptic cell, it will travel down the axon, and then here, right, this would interact with the postsynaptic cell. And again, what we're seeing here is you have this myelin sheath. And so if you notice, if you kind of blow that up, that myelin sheath, this is really acting like insulation, if you will. Okay, so just like if you have a wire and you have insulation wrapped around it, it really goes ahead and speeds how fast that electrical impulse kind of travels through it. Same thing kind of here, right? So the current will travel faster in this case. So you have this insulation kind of around it. One thing to kind of know, right, is if the diameter of the axon increases, right, so as you go from here to here, the signal will travel faster, okay? And then also, right, if it's myelinated, right, so if it's myelinated, if it has this myelin around it, that will also allow it to travel faster because it has insulation. And we'll look at later, it also allows you to kind of have what we call saltatory conduction, where you're going to go ahead and generate action potential only at these nodes and not have to generate it all the way down the axon. So summarizing this, right, it's going to travel, the information is going to travel down the axon right from the presynaptic cell as the signal is received on the dendrites, and then it's going to go ahead and be transferred to that postsynaptic cell. The myelin, again, is acting like insulation, and it's allowing the signal to move faster down the axon, right? And if the diameter is larger for the axon, the signal will also travel faster. And something to also take a look at right here as well is, so once it arrives, so again, this would be that presynaptic neuron. And once this information, once this signal arrives here, as you can see, there's these synaptic vesicles, okay? And we'll look at this in more detail later, but essentially once that signal arrives, it's going to cause calcium to enter, and as calcium enters, that's going to trigger the synaptic vesicles to go ahead and fuse with the membrane and release their neurotransmitters into the synaptic cleft, which they can then bind right on the dendrite of the next one. Okay, so on the ligand-gated channel, right? So what we're seeing, right, is we can send, a big thing to focus on is, right, we can send information through electrical signals, right, which is what we're seeing sent down the axon. And then we can also send information, right, this message through chemicals, which is what we're seeing at the synapse, right? Because there's chemical synapses and there are also electrical synapses as well. Okay, but this one that we're seeing here, this is a chemical synapse. So this is going to allow us to start thinking about generating an action potential, right? So as you can see, the membrane potential, right, so meaning the difference between both sides of the membrane, in many cells, in many axons, or in many nerves, is somewhere between -70 or really -80, right, and about -60. So it tends to lie somewhere between there. And then really, as we'll look at in a bit, so as you can see, as it becomes depolarized, so as we'll look at as we're generating action potentials, as the neuron, well, as the difference becomes more positive, so there's less of a difference, it will go ahead and generate this action potential, allowing us to send this electrical signal down the axon, right? And then we'll look at repolarization and then how kind of resting potential gets regenerated. But here's where we'll focus first. We'll focus on what does the cell kind of look like normally when it's at rest, when no signal is kind of being sent. Okay, so if we're looking at, this is what it's looking at resting potential. So here is the outside, right, and this is the inside, okay, of that neuron. And so if you can notice, right, outside, you can very clearly see that there is a lot of sodium. So the circles, these are sodium ions, okay? And what you're seeing here, right, is these squares, these are potassium ions, okay? So there's more, in this case, right, there's going to be more sodium outside the cell and more potassium inside the cell. Well, in general, what's happening to generate a resting potential kind of at this point is the fact that what we're going to end up with, right, is that the inside of the cell is more negative. Okay, so the inside of the cell is more negative because again we said at a resting potential we're about at a -70 millivolts. Okay, so that it's more negative inside compared to outside, right? And it's more negative inside because you have very large negative molecules such as proteins that are trapped in here. And then what we're also seeing at this point is, as you can see, right, what happens for the most part is sodium, so you have these sodium channels, right? So here's the sodium channel. You also have potassium channels, okay? And in this case, right, the sodium channels are often allowing very little sodium in, right? So there's very little movement of sodium inside the cell, so very little movement of positive sodium ions in, but there's actually more movement of these, right, of the potassium ions out. And so that's one thing, right? As the potassium goes out, that is also going to make the inside more negative. Okay. Another thing is too, is if you notice over here, this is what we call a sodium-potassium pump. And what we're seeing here, right, is actually if you notice, okay, what you're seeing is there are 3 sodium ions that get sent out basically for every 2 potassium ions that get sent in, right? So 2 potassium ions are pumped in and 3 sodium ions are pumped out, okay? And if that's the case, right, what you're also seeing is that since there are 3 sodium ions that are being pumped out for 2 potassium ions that are pumped in, right, this is also going to make the inside more negative. Okay, so both of these mechanisms, right, both of these things are helping to make the inside so that it has a more negative, right, so that it would be more negative compared to the outside, which is more positive. Okay, and so this is what the resting potential of a neuron looks like, and this is how a resting potential is generated. So let's go ahead and take a look at an animation that's going to help kind of summarize this and allow us to look at the resting potential a little bit more.
A resting neuron, one that is not conducting signals, contains potential energy resulting from an electrical charge difference between the inside and the outside of its plasma membrane. A resting neuron, one that is not conducting signals, contains potential energy resulting from an electrical charge difference between the inside and the outside of its plasma membrane. The cytoplasm just inside the membrane is negative in charge, and the fluid just outside the membrane is positive. The membrane is said to be polarized. Like a flashlight battery, the difference in electrical charge carries potential energy, which can be measured as a voltage. The oscilloscope shows this charge difference, called the resting potential, in millivolts. A neuron has a resting potential of about -70 millivolts. What causes the resting potential?
The plasma membrane of the neuron keeps large negatively charged molecules such as proteins inside the cell. Pumps and channels in the membrane regulate the flow of positively charged sodium and potassium ions. The neuron maintains a higher concentration of sodium ions outside the cell than inside, and a higher concentration of potassium ions inside the cell than outside. In a resting neuron, most sodium channels are closed, but many potassium channels are open. As a result, very little sodium can diffuse into the cell, but potassium ions can flow freely out of the cell, adding to the positive charge outside. The sodium-potassium pump actively transports more sodium ions out of the cell than potassium ions into the cell. This contributes to the buildup of positive charge outside the cell, while keeping the concentration of sodium low and the concentration of potassium high inside the cell. The diffusion of positive potassium ions out of the cell and the large negatively charged molecules trapped inside the cell cause the charge difference that constitutes the resting potential.
Okay, so now that we looked at resting potentials, right, which is how the neuron is starting, let's go ahead and look at how we can generate an action potential. So when we're going to generate an action potential, which we'll look at in a lot of detail, the membrane is going to become depolarized. It's going to become more positive. We can also see, just before we start talking about action potentials, you can also see it become hyperpolarized, right? So this would actually mean that it would be becoming more negative, right? So it's becoming more negative. It's what we call hyperpolarized. And this is the case when you get an inhibitory signal, right? Because what we're seeing when we're going to generate an action potential, that's some sort of excitatory signal, right? That's going to allow us to go ahead and send a signal down the neuron. So in this case, right, what we're going to look at is action potentials, where it's become depolarized and that we're going to send a signal down the neuron. Whereas here, right, if it's hyperpolarized, that's making it more negative. It's making it harder, right, to reach the threshold, harder to go ahead and generate an action potential. So that's just kind of something to think about as we start. So when we're going to generate an action potential, the first thing that's going to happen, right, is so if you, right, here we are at our resting potential at like -70, okay? And what we're going to do is we're going to receive like some sort of excitatory signal, right? So you're receiving a signal, okay? And as you're receiving this signal, what happens, right, is that this signal is going to go ahead and you're going to see sodium ions will start to enter, okay? So sodium ions start to enter, and what's going to happen right at this point is some sodium ions will start to enter, and that's going to go ahead and it's going to start to change the voltage, okay? It's going to start to change the voltage that you're seeing, right? So you see that the voltage is increasing, and it's increasing to reach this threshold. This threshold is about -55 millivolts, okay? And so it's reaching this threshold, okay? What we'll end up going ahead seeing happen at this point, right, is, and as it's reaching this threshold, right, what's happening is, as it's reaching the threshold, what we're going to see happen is the action potential will get generated, right? And so this is kind of an all or nothing response, right? So once you reach the threshold, it's an all or nothing response. We're going to go ahead and we're rapidly going to depolarize the membrane. And how does that happen? Well, that's going to happen obviously because now this more sodium ions are going to go ahead and enter the cell, okay? And these voltage, so what's also important to remember is that these sodium, so you have these sodium channels, right? But that are there otherwise, but you also have what are called these voltage gated ion channels. So you have this voltage gated sodium channel, okay? And with this voltage gated sodium channel, right, once the appropriate voltage is reached, they start to open, right? And so as many of these start to open, we're very quickly going to go ahead and generate our action potential, right? Because lots of sodium ions are coming in. Okay, so lots of sodium ions are coming in. There are also potassium channels, but as you can see, right, these potassium channels are closed, okay, at this point. And since the potassium channels are closed and the potassium staying inside, right, it's becoming much more positive inside. Okay, and so now, right, you're actually at a point where the inside is more positive than the outside, which is negative, right, which is why you can see that the action, with the action potential, we're above 0 millivolts, right? It's actually more positive inside compared to the outside. Okay, well, then what happens, right? At that point, okay, what you start seeing happen is now what happens, right, is these voltage-gated sodium channels, they get inactivated, okay? And as they get inactivated, right, the sodium no longer is coming inside. And then what actually happens at that point is these potassium channels open and potassium ions start to rush out of the cell, okay? And as potassium ions start to rush out of the cell, so since no more sodium's coming in, right, potassium's rushing out, and as potassium's rushing out, this is making the inside less positive, right, or more negative, if you will. And so that's why we're seeing, right, the falling phase of the action potential, right, where we're starting to see this go back down, okay? And then what we actually see, right, is that we get this kind of undershoot, if you will, right, which is what we're seeing here, okay? And with this undershoot, what's happening is you still have, right, the sodium ions not entering, and you have the potassium ions leaving, and it actually goes under the resting potential. So you actually get more of the potassium ions that go out, and which is why it goes below where it was at the resting potential. And then what actually happens, right, is that you have, if you remember, let's go back. If you remember, you had these sodium-potassium pumps, right, that would go ahead and they would take 3 sodium ions and move them, right, whereas it would move 2 potassium ions, okay? So that as you were looking at these 2 pumps, right, so, and what would happen, right, at that point is it would go ahead and pump 2 in, right, and 3 out. And by doing that, you're going to go ahead and this would allow you to kind of reset your resting potential. Okay, and so also it's going to reset the concentrations, right? So because again, you started with a lot of sodium outside, right, and then a lot of potassium inside, and these pumps are going to allow you to go ahead and reset that. Okay, so that's what's going to happen in an action potential. So let's go ahead and take a look at an animation, which will go ahead and give us a little bit more information.
We've looked at the resting potential in a neuron that is not transmitting nerve signals. What happens when the cell is stimulated? A stimulus may change the permeability of the cell membrane to sodium or potassium ions. This alters the distribution of charge in the cell body, and if the change is great enough, it triggers a signal called an action potential that moves down the axon. How does the action potential move along the axon? Voltage-gated channels in the membrane open and close in response to voltage changes across the membrane. When no action potentials are being transmitted, most of these channels are closed. An action potential begins when voltage-gated sodium channels open and sodium ions rush into the cell. The plasma membrane becomes positive on the inside and negative on the outside. Very quickly, the sodium channels close, while voltage-gated potassium channels open, allowing potassium ions to rapidly diffuse out. The plasma membrane returns to being positive on the outside and negative on the inside, and the potassium channels close. Meanwhile, the sodium ions inside the cell have diffused to adjacent areas, causing a slight change in the polarity of the membrane ahead of the action potential. This change in polarity causes the voltage-gated sodium channels along this part of the membrane to open. Again, sodium ions rush in, and the action potential spreads to the adjacent part of the neuron. In this way, the action potential travels down the neuron like a wave. The efflux of potassium ions during the falling phase of the action potential restores the negative charge inside the neuron. Meanwhile, the sodium-potassium pump has been shuttling sodium ions out and potassium ions in, re-establishing the resting distribution of sodium and potassium ions.
Let's look at another action potential as it moves along the membrane. This time we will measure the voltage changes that occur in one spot along the membrane. A slight change in polarity causes the voltage-gated sodium channels to open. Sodium ions rush in, causing a reversal in membrane polarity. Sodium channels close and potassium channels open. As potassium ions rush out, the membrane returns to being negative on the inside. The sodium-potassium pump then restores the balance of ions present at resting potential.
Okay, so now we can see that the action potential is actually conducted down the neuron, right? And as we saw kind of in the animation, what you're seeing is that, again, as the sodium ions come in, it's causing the depolarization, okay? And so that action potential is generated. And then the fact is, right, that causes a local depolarization, which causes that to get kind of propagated down the axon, right? And so you can see, right, that as we're moving here, right, as we're going ahead and moving down the axon, you're seeing that the action potential moves down as well, right? And then again, those potassium ion channels are opening, and so then as potassium's rushing back out, it's kind of restoring that resting potential, right? And then we're also going ahead and using the sodium-potassium pumps to do so as well. So what we're seeing again, right, is where that signal is being sent from that presynaptic neuron to the postsynaptic neuron, and that's traveling down the axon, right? And so you're getting this action potential moving down it. And again, back to this idea of myelin. So myelin, again, is acting like this insulation. So first, right, so if I have a larger diameter axon, the signal is able to travel faster. It's essentially like having less resistance, okay? And then also as we're looking at myelin, myelin again is acting like insulation. Now the other thing that's important too essentially is with myelin, you get what's called saltatory conduction, where basically it's going to go ahead and jump, right? It's going to jump to these nodes of Ranvier, okay? And so what we're seeing is that it's going to jump because it only has to generate an action potential at each of these nodes. So it will generate an action potential here, okay? And then that will be enough to stimulate and generate it here and generate it here. And since you don't have to generate it at each little spot along the neuron, it's able to travel much faster. So that's another reason why the myelin is going ahead and how it's increasing really the speed of conduction of that signal. So once it goes ahead and it reaches the synapse, right? So we'll talk about the synapse in a little bit more detail. So here the signal is going to go ahead and reach the synapse, okay? So now at this point, the action potential has reached the synapse. And so here's our presynaptic neuron. And what happens, right, is it goes ahead and reaches the synapse. And since it's going to reach the synapse, calcium ions are going to go ahead and enter. And as calcium ions go ahead and enter, they're going to cause these synaptic vesicles, right, to fuse with the membrane, of the presynaptic neuron. And as they fuse, they're going to release their neurotransmitters, right? So they're going to release their neurotransmitters into what's called here is this space, which is called the synaptic cleft, okay? And then what they're going to do is they're going to go ahead and they're going to bind on these channels. And these are ligand-gated ion channels. And they're going to go ahead and bind. And what will happen is they'll bind to this, and they're going to allow now sodium ions to enter. So sodium ions can go ahead and enter on this postsynaptic neuron. And as sodium ions enter, right, and also some potassium ions can leave, right. So if this signal is strong enough, right, so that you're getting enough sodium ions entering, right, because that is the goal essentially, if it's excitatory, right. So if the sodium ions are going to go ahead and enter, then what will happen at that point is that will cause a depolarization on the postsynaptic neuron, right? And if that's depolarized, then we can go ahead and we can generate another action potential, okay? And if we can generate another action potential, then now we're going to send the signal down the next neuron, okay? And so this is a chemical synapse, right? So this is a chemical synapse because we're using these neurotransmitters.
Action potentials arriving at the presynaptic terminal cause voltage-gated calcium ion channels to open. Calcium ions diffuse into the cell and cause synaptic vesicles to release acetylcholine, a neurotransmitter molecule. Neurotransmitter molecules, acetylcholine, diffuse from the presynaptic terminal across the synaptic cleft and bind to their receptor sites on the ligand-gated sodium channels. This causes the ligand-gated sodium ion channels to open, and sodium diffuses into the cell, making the membrane potential more positive. If the membrane potential reaches threshold level, an action potential will be produced.
So let's take a look at some of the neurotransmitters that are released into the synaptic cleft. One of the ones that was mentioned in the animation was acetylcholine. Right? So acetylcholine is a really common neurotransmitter. As you can see, it's involved in muscle stimulation, memory formation, learning, and there's a number of toxins that actually disrupt acetylcholine. One example is sarin gas. We also have, right, so you can see kind of looking at structurally, there are a couple of amino acids that are there. So these are a lot of neurotransmitters. There's a bunch of other ones. GABA is one. It's an inhibitory one, right? So that would cause hyperpolarization. An example of a drug is Valium, which reduces anxiety by binding to that GABA receptor, right? So it's hyperpolarizing it, it's not allowing you to send signals. Whereas glutamate is excitatory, so it's going to cause action potentials to happen, and it's the most common neurotransmitter in the central nervous system, also involved in memory formation. Then we can see norepinephrine, right? And that is most commonly released to affect the heart, right? And it's increasing heart rate. And you can see dopamine and serotonin affect sleep, mood, and then actually, interestingly enough, Parkinson's disease is associated with a lack of dopamine. Endorphins, right? Those are something that increase your pain perception. So if you have a high rate of endorphins, you're able to kind of sense a greater amount of pain. And then lastly, there are some gases that are released, right? And nitrous oxide is one interesting one, right? And that's made on demand, and it causes smooth muscles to relax and blood vessels to dilate, right? And this is an example of a gas that's also acting kind of as a neurotransmitter. So these are a couple of examples of neurotransmitters that are going ahead and being used in the synapse, right? And so now we're going to go ahead and wrap this up with a final video that's going to go ahead and show kind of a nerve impulse.
A nerve impulse is an electrical current that travels along dendrites or axons due to ions moving through voltage-gated channels in the neuron's plasma membrane. Voltage-gated channels open and close in response to an electrical voltage, so they are affected by changes in electrical charge around them. When a neuron is at rest, a charge difference is maintained between the inside and outside of the cell. This charge difference is produced and maintained largely by active transport using sodium-potassium pumps. The pumps send sodium ions out of the cell and bring potassium ions in. While other channels allow some flow of potassium ions back out of the cell, the sodium ions cannot easily get back in to replace the lost positive charges. The overall result is that the exterior of the cell has a net positive charge and the interior has a net negative charge. The difference in charge between the interior and exterior of the cell is called the resting membrane potential. A nerve impulse begins when a stimulus disturbs the plasma membrane on a dendrite, causing sodium channels to open. Sodium ions flow into the cell, lessening the charge difference at that location. If the change is enough, it will cause nearby voltage-gated sodium channels to open. This allows so many sodium ions to flood into the cell at that location that the membrane there is depolarized, with the local region inside the cell having a net positive charge and the outside of the cell having a net negative charge. This affects neighboring voltage-gated sodium channels, which then open, moving the depolarization along the membrane. This moving depolarization is called an action potential. Changes occur behind the action potential to restore the resting membrane potential. The voltage-gated sodium channels close and voltage-gated potassium channels open. This allows a rapid flow of potassium ions out of the cell, repolarizing the membrane so that the inside is again negative and the outside positive. This is followed by use of sodium-potassium pumps to fully restore the resting membrane potential and to reestablish proper concentrations of sodium and potassium ions inside and outside the cell.